where I would take a drink of the drink I brought this morning and forgot to bring with me. Everything else just got in the way. All right, let's look at the citric acid cycle. So this is the first stage we're looking at where we regenerate something in the process. So acetyl-CoA, or some people pronounce it acetyl-CoA, either one's fine. Acetyl-CoA is going to go into this, and we're specifically following the movement of these two carbons, which then leave as carbon dioxide. And then in order to generate some ATP and some of the electron carriers, there are some stages of this cycle that bring around the starting material back to where we started. Okay, some of you have potentially learned this as the Krebs cycle. That's the same thing. That is the tribute to the biochemist who discovered most of the reactions. All right, so let's look at this piece by piece. Remember, this is happening inside the mitochondria, okay? Let's see, do I have maybe we can look at this one. It looks complicated because it is, but remember, we're focusing just on a few aspects of it. If you take biochemistry or cell biology, I believe you'll probably maybe memorize a lot of this, actually. I did in my biochem class as a student, so I suspect you might as well. But we're not going to be that ambitious. We want to know what goes in, what comes out, and what is the result. So let's think about it in those terms. So what goes in, these are the two carbons from our formerly pyruvate molecule, now acetyl-CoA. Those two carbons are joined to a 4-carbon molecule called oxaloacetate down here, and that gives us our 6-carbon molecule. This is not glucose, but it does have 6 carbons. Remember those two little carbons that we just put in the acetyl-CoA, excuse me, are going to leave soon. So let's mark them off as special. We might say that the oxaloacetate is backpacking those carbons until they can be released. I think that's the favorite verb that I ever heard my son use when he described his soccer team, which had one player that scored 90% of the goals. And he said, I can't remember the player's name, but he said, that guy's been backpacking our team all season. It's such a descriptive term. Is that used in other contexts? Have you heard that?
Yeah.
Okay, so that.
Or piggybacking too.
Okay. Yeah, I thought that was really funny. He's been backpacking this team all season. It was true, but anyways. Okay, so the citrate is going to backpack our little acetyl-CoA carbons until we break them off and start to release electrons. So when covalent bonds are broken, electrons are released. Those electrons have to go somewhere, and that's where our electron carriers come in. Okay? Soon we are going to strip off one of those carbons as carbon dioxide, which means more electrons have to be collected. So there's another NADH, another electron carrier. Then we're going to release more carbon dioxides, and now all the carbon dioxides that you consumed as that sugar molecule have been recycled. Okay, so those go into the atmosphere. The plants are going to take those carbon dioxides, make sugars with them. We're going to eat the plants or eat something that ate the plants, and it's going to go full circle. Okay, after that point then, the rest of the citric acid cycle is doing the business of generating ATP. Not much though, right? For all these reactions, only one ATP for every acetyl-CoA is made, but that's not the point. The point is that we are going to strip all of these high energy electrons off until we get back to the oxaloacetate where we started. Okay, so these are the key things to remember. We start with 2 carbons, we release 2 carbons, so now we've broken even. Along the way, as we break and rearrange bonds, electrons are released that get captured by electron carriers, and at one point we have unstable bonds that generate an ATP. Okay? All right, so let's go back to the big picture. So now we don't have any carbons from our original molecule left. All we have are electron carriers. And keep in mind that not all of those electron carriers are going to be used for the process of generating ATP in the last stage. Some of those NADHs are used for other things in the cell. So that's why, and I probably talked about this last time, that's why we have a range of potential outcomes. we should, if it's a chemical reaction, be able to say there will be exactly 32 ATP made, blah, blah, blah. But we can't say that because this is not a closed system. All of these electron carriers are not always going to be used in this process. The cell has other plans for some of those guys. So we'll use most of them, but not all of them. Therefore, we have that range. All right, now, part 4, oxidative phosphorylation. This is probably the coolest part. And in this diagram, it's a little hard to tell. Let's see. What is, I mean, you can maybe look at the top here and figure this out, but what is not as clear is that this is all inside of the mitochondria. So I'm going to draw the picture that is not shown. It's a pretty good mitochondria, if you say so myself. And then this would be the cytosol out here. And then this, could be our cell. Let's give it a little nucleus. And a lysosome. All right, so everything that's happening within the mitochondria, the way it's drawn here, it looks like this stuff is going out of the cell or into the cytosol. It's not. All of this is happening in this little space. And again, this would be nice if this was a bigger diagram. All of it's happening in that little space in between the outer and inner membranes. So in that space, remember, ions, like the charged hydrogen ions, which we'll just call protons most of the time, they cannot cross membranes. So that means if we pump them into a space surrounded by membranes using some type of transporter, they can't cross back unless they're given a way to cross back through a protein. So everything is kind of, I don't know what the right word is, airtight, watertight. Those hydrogen ions, if they're pumped out there, will create a gradient, and then you have to give them a way to get back in. Okay, that's a key thing. So the oxidative phosphorylation is sometimes called the electron transport chain because electrons are going to be moving along this pathway. I guess I shouldn't have used red. My red is now overshadowing the other red. How about we use yellow? So the electron transport chain, which is not very obvious since I used red to make all those lines, is moving along here until it gets to there. It's even worse, isn't it? I just made it worse. Oh well, story of my life. But watch where all the electrons come from. So we have NADHs up here, and remember, NADHs started as NAD+. Right? NAD+ was the electron carrier before it had the electrons. When we add the electrons, it becomes NADH. So now we're reverting back to our starting material. Same with FAD. This was pre-electron carrying mode. Once we add electrons, it becomes FADH2. So both of those are going to be giving these electrons away. That was the point, right? The point of an electron carrier is to be temporarily modified by taking on high energy electrons and then give them away to the correct recipient. So the recipient is either NADH dehydrogenase or a molecule called ubiquinone, usually just abbreviated as Q. So both of these guys receive the electrons, and the electrons are moving through the membrane on a path between multiple stages. And if you notice, 3 of the 4 destinations for these electrons are pumping hydrogen ions. Okay, so in the electron transport chain, we have these protons, or we have these proteins, which are called proton pumps. All right, and the first one, where NADH is passing along its electron, is called NADH dehydrogenase. It's a mouthful. All right, and if you think about the name there, dehydrogenase, it sounds like it's taking a hydrogen from NADH, which is why it's called that. That's what it's doing. So the NADH donates an electron, and in the process, it also donates a hydrogen, and that hydrogen gets pumped across. The second proton pump, is called cytochrome BC. And cytochromes actually have a pretty interesting, well, let me back up. Let me finish my story here. The third electron pump, which is the end of the road for this high energy electron. So keep in mind, this same electron is traveling this path. So once NADH and FADH2 give these electrons away, those electrons are moving down until they find oxygen. Okay, so the last one is called cytochrome oxidase. And collectively, these proton pumps use energy from electrons to pump hydrogen ions into the intermembrane space. This is the space between the membranes of the mitochondria, as the name implies. Now, a little sidebar is that this little tangent of the electron's journey, where cytochrome C carries the electron from one pump to the next. Cytochrome C, if you are interested in the relationship between species, so this seems completely unrelated to this because it is, but this molecule, cytochrome C, is used by zoologists and evolutionary biologists to determine the relatedness between organisms. And the reason is that, well, at some point someone just decided, let's use this gene. And it's a gene that's encoded by mitochondrial DNA. So remember, a weird thing about the mitochondria is that it has its own DNA. And if you sequence the gene cytochrome C from one organism, it will have a specific DNA sequence. And if you sequence the DNA from a very closely related organism, it will have a similar but not exactly the same sequence. And the closer those two sequences are to each other in similarity, the more closely related the species are said to be. And so once someone chose this gene as kind of the hallmark gene to use across other species, people started sequencing the cytochrome C gene from all kinds of organisms, from all steps of life. And if you see what's called a phylogenetic tree, which usually looks something like it looks something like this, where there is an assumption that there is a common ancestor from which all organisms are derived. And so on the phylogenetic tree, if you see, like, for example, we are most closely related to chimps. It's like 99% or so DNA similarity. And then down here, maybe we have other primates. I don't know. All of this is just hypothetical. I know we're on the same branch as chimps. What's another good primate? Orangutan. Have you read Murders in the Rue? Who said that? Murders in the Rue Morgue. It's an Edgar Allan Poe story. How do you spell orangutan? Does it have a G at the end? No. That feels wrong. That feels like a drink. If I take the G away, am I right? Closer? Okay. Anyway, if you are into gory stories, which I'm sure you are, till death do us part.
Yeah, you had it right.
In the story, they can't figure out how a guy is murdered because someone broke into the balcony and then he left through the chimney. And they realized eventually an orangutan got out of the zoo, ripped this guy's arms off, like dragged him through the chimney and murdered him. It's a touching story. Anyway, all of that to say, if you look at phylogenetic trees, often they are, strangely, the result of studies where they use a certain molecule, for whatever reason it was chosen, and they compare the sequences, and then they arrange the individuals on the phylogenetic tree based on how similar that sequence is. So this is a good improvement over just using morphology, because people used to just use structures. They used to say, well, they look similar enough, they have the same bone structure, etc. But there's something you probably know called convergent evolution, where two different things can be similar, because they've adapted to similar environments, but it doesn't mean they're related, right? Bats can fly, but they're not birds, right? So anyway, let's keep moving. All right, so we've got our proton pumps. They use energy from electrons to pump hydrogen ions into the intermembrane. Oops, I forgot my last word. Intermembrane space. Okay, and the end of this story about the proton pumps is that they create an electrochemical gradient. Okay, this is the second time we've seen that. Electrochemical gradient. The electrochemical gradient means that you have a difference in concentration of a molecule on one side of the membrane versus another, and you have a difference in charge. So the place we saw this previously was the sodium-potassium ATPase, where we pumped out sodium ions and we pumped in potassium ions, and both of those ions carry a single charge, but we pump out more sodium than we pump in potassium. And so we create an uneven gradient of both molecules and charges. So similar concept, but here we have multiple pumps pumping hydrogens out using the electron energy. Now, the reason this is called oxidative phosphorylation is that at the end of it all, we have oxygen and we have phosphorylation. The phosphorylation is going to be adding a phosphate to ADP. So that's the whole point, right, to get that ATP. And the oxidation is that these electrons eventually end up joining oxygen to produce water. Okay, so without oxygen, none of this can happen, which is a strange thing to me. In my mind, I would think without oxygen, you can go through all the stages, you just can't do the very last thing. But that's not how the chemistry works. The way the chemistry works is without oxygen, you only do the first stage. You don't even bother with citric acid and pyruvate breakdown. You just do glycolysis, okay? And so in the case of, well, let me define another word. One of the important terms that's emphasized in your book and emphasized in this process is the term chemiosmosis. Okay, chemiosmosis means, the production of ATP using energy from an electrochemical gradient. The production of ATP using energy from an electrochemical gradient. And so that is what's happening in this process. So chemiosmosis describes the kind of the final event where ATP is produced because we've made an electrochemical gradient. Along the way, we want to make a note that oxygen carries an important title here. I wonder if they say it in here. No, they don't. I thought they did. So oxygen is called the final electron acceptor. Bless you. With electrons and hydrogen ions, the oxygen forms water. So to kind of put a big picture spin on this for just a second, since we're talking about the oxygen forming water at the end, one way that people describe respiration, and this isn't going to be a balanced chemical equation, this is just going to talk about inputs and outputs, is that we have glucose plus oxygen yields ATP and carbon dioxide and water. Okay, so if oxygen is present, we would call this process aerobic respiration. Now, if you look at that for just a minute, glucose and oxygen are the inputs, the reactants. ATP and carbon dioxide and water are the products. Do the components of those reactions mean anything to you? Do they look like anything else you know of? Anything else in that reaction look familiar?
Oh, photosynthesis? Plant? You drawing a plant?
Yes. You beat me there. Yeah, so finish your thought. What do you mean photosynthesis?
Isn't that the equation?
If we go backwards, right?
Oh, wait, yeah, that's, cellular respiration, and if you go backwards, it's photosynthesis, because the reactions for one are the products for the other one.
Yes.
And that's why you have to do them in,
That's why we
but they
have
go hand in hand.
to live with plants. Because plants use carbon dioxide and water and a ton of ATP to produce glucose and oxygen. And photosynthesis also relies, of course, on energy from light, from the sun or from grow lamps, I guess. Okay, so that's why oxygen is important. If we don't have oxygen as the final electron acceptor, none of this happens. Okay, we don't get past glycolysis. All right, so let's take a look at this again. Here's our electrochemical gradient. And as the electrochemical gradient forms, the hydrogen ions, or the protons, need to come back across the membrane. Well, they don't need to, they want to. So as this gradient gets more and more concentrated, the hydrogen ions are going to be looking for a back door to get back in. And that back door is this massive enzyme called the ATP synthase. Okay? And so maybe we'll make an addendum here. The production of ATP using energy from an electrochemical gradient, and then let's put a little asterisk there. The protons return to the matrix, and remember the matrix is the component of the mitochondria in the center. So this is our, I don't know if it really, well it does say it in the very small word there. This is our mitochondria, the inside is the matrix, and in between is the inner membrane space. Okay, so the protons return to the matrix via, if I may use that word, an ATP synthase enzyme. Okay, so this enzyme is both a transporter that allows hydrogen ions to cross, and it's enzymatic in that it synthesizes ATP. And so as the protons return to the matrix via the ATP synthase enzyme, energy from their transport meaning when they come back across, they bring actually mechanical energy. Energy from their transport powers the production of ATP. So this is where we have been driving for this whole time, to package those high energy electrons to the mitochondria, to release them to the proton pumps. The proton pumps use that electron energy to move hydrogen ions against their concentration gradient into the inner membrane space. This creates an electrochemical gradient, which powers the chemiosmosis, the production of ATP using energy from the gradient. And the thing that catalyzes this all is the ATP synthase pump, which takes ADP and phosphates. And as those hydrogen ions come across, I don't think I have a closer picture of that. No, I do have a better picture of that. There's actually physical movement of this pump. And so as the hydrogen ions come across, this actually turns. And as it turns, it pulls two molecules together, namely the ADP and the phosphorus. So this transfer of mechanical energy is used to form a chemical reaction by bringing two things that want to interact close enough together that a covalent bond forms. And there's actually, I think it's your book that talks about this experiment where people actually linked a protein to the ATP synthase pump that was long enough and big enough that you could actually visualize it with a microscope. And what you could see was that it was turning. As ATP was being made and hydrogen ions were coming back across, you could actually see the physical movement of this enzyme, which is pretty wild. All right, let me see if there's anything else I want to say about this. I don't think so. Okay, and then the diagram here, which is useful but mostly conceptual, is that as electrons move from the electron carrier to one pump, then to the ubiquinone, which carries it to the next pump, then to cytochrome C, which carries it to the next pump, and then finally to oxygen, which makes the water, the energy goes down. So that original electron with the high energy harvested from the glucose and the pyruvate and the acetyl-CoA, that high energy is going to diminish. And I don't know how they measure this. I'd love to know. I don't have the chemical sophistication to know how they do it. But the energy is going to diminish as it travels eventually to its final electron acceptor, oxygen. And they put a half O2. That's a common way to write oxygen into an equation because oxygen never exists as just a single oxygen atom. Okay, you'll never see a single oxygen atom just hanging out, but you will see oxygen gas. And so to balance the equation, 2 hydrogens and 1 oxygen to equal water, you say half of O2, because prior to being the other part of the water molecule, that oxygen was existing as oxygen gas. All right, let's now look back at our diagram. We're about to peak. We're not too early. It's about to happen. Okay. Bucky's gonna join us in our learning experience. Cut the board, Bucky. Just one moment, I need your attention. All right, so the glucose breaks down 2-pyruvate with 2 NADH. Those are the electron carriers, 2 ATP. We know that the pyruvate then breaks down again, yielding one of the carbons from that 3-carbon molecule is carbon dioxide. The other 2 go into the acetyl-CoA. The acetyl is the 2-carbon group. The CoA is that other long, beautiful molecule that hangs onto it for just a moment. Then we have the production of, from the 2-carbon acetyl-CoA, 2 more carbon dioxide molecules, and then we gather all these electrons in NADH and FADH2, and then we have production of ATP. And then all of those electron carriers eventually yield ATP molecules. Let's think about this. Oh, don't go there yet. Not gonna peek too early. Oh yes. Yep. It's happening. There are no consequences to this experiment. Doesn't matter if you get something wrong, but just looking at that. The question that we have before us is this. If you close your notebooks and we turn off the slide for a minute, just try, see what you can fill in. Again, I'm not grading it, I'm not taking it up, there are no consequences. This is a test of what you know so that you can have sort of a self-assessment. Just take a moment. Relax. There's no pressure. See what you can do, and then we'll go on from there. Just try. That's all we ask. the second one, and then when you try, you can fill out the correct from the slide. But I don't have one. I don't know where did our stray sheets go? Try to think logically, use context clues. Remember, the arrows mean something. When arrows come from one level down to the next level, it means those things are descended from the previous molecule. If molecules are made that don't carry on to the next level, that don't have an arrow going to the next level, those are molecules that are probably going to be either ATP or electron carriers, as opposed to pyruvates, acetyl-CoAs, glucose. Follow your heart. It's never let you down, right? Imagine a world where grades didn't matter. That world's just not real. Okay, let's see if we peaked too early. So take a look at it again. Look at where your strengths are and where your flaws may be. And again, keep in mind, wherever we have the dotted lines, those are going to be ATPs. That's the way I've set it up. So if you can knock that off of your your list early, go ahead and put in all the ATPs. And then remember that when a molecule breaks down, in the first to the second stage and the second to the third stage, carbon dioxide is going to be made. So we'll have CO2 in both of those stages. And then molecules that don't go on to make more molecules in the next generation are going to be electron carriers like NADH and FADH2. So we'll see that at every level we have NADH, and at the last level only we have FADH2. And we did, I don't have it written in the stages here, but you guys seem to know that pretty well. First stage glycolysis, pyruvate breakdown, citric acid cycle, and oxidative phosphorylation. I'll give you a second to correct that so that you have a good copy of that. Of course, this is available to you on Blackboard as well, so you need to go back and look at it. Oh, yes.
you put down Krebs instead of citric acid cycle? Is it still correct?
I will celebrate your tribute to the founder of those reactions. Definitely correct to put Krebs or citric acid cycle. Both are very much accepted. Okay, how are we feeling about this now? Did we peak too early, or are we
I think I'm in a valley.
in a valley? That's not good. Well, the good news is we didn't have to peak today on this. This was a self-assessment to see if we will be peaking next week. Let's see. Let's make our aerobic respiration quiz.
Is it just this chart?
That sounds right.
He's being nice to us.
I am being nice to you. How much clearer, more explicit could I be? Okay, so put that away for now. Let's let your break. Let's look at a couple follow-up items to aerobic respiration. But now you've seen and experienced what the quiz will be like, and I have full confidence that you are going to hit a home run on this one, which will give you a boost going into exam 2. All right, so we have just a few minutes left here, a little bit of time. Let's look at a few big picture things, and then we'll be able to finish up early on Thursday with this material, and then we can either start reviewing or I might introduce a new topic. We'll see. I'll give it some thought. So far, we have been operating under the sort of mode that, oh, I can't do yellow. That doesn't feel right.
You should do blue.
I want to do purple. I'm feeling purplish. What's wrong with that?
I think you should be doing purple.
I do feel purple. So we've been operating under this assumption so far that our input was a carbohydrate like a glucose, that we went through glycolysis, then we went through pyruvate breakdown to acetyl-CoA, then we went through the citric acid cycle, and then oxidative phosphorylation. But let's think about the fact that you are not on a 100% sugar diet. I don't think. Maybe you're close. Hopefully not. But you also eat proteins, and you also eat fats. It'd be impossible to avoid those in a college food setting. And so what happens to those things? Well, keep a few things in mind. First of all of those things, proteins, carbohydrates, fats, as well as, it's not listed here, but nucleic acids are in everything you eat, right? If you eat meat, there is DNA in meat. Right? That DNA came, or that meat came from an animal. That animal, like you, has DNA. So when you eat meat, you're eating DNA. When you eat plants, you're eating DNA, right? It's in there. In fact, you can isolate DNA from cooked foods. And I know this because in the genetics lab, we isolate, just like you will at the end of your semester, isolate DNA from your cheek cells. You'll swab your cheek with a toothpick and you'll isolate DNA, you'll do a little experiment with it, and so forth. But if you had, for example, some leftover meat in your mouth, let's say you just ate a chicken sandwich right before you swabbed your cheek, that meat will contribute DNA to the experiment, right? Because it has meat. You can test whether or not a plant was produced by a biotech. I'm not phrasing this well. Let me redo that grammatical train wreck. You can tell if food is, what's the word? Yes, bioengineered. I could not put my hand on that. You can tell if a food is artificial in that sense by whether or not it has certain types of DNA in it. So people can test food using a DNA test to see if it came from, for example, Roundup Ready corn, right? That's corn that has an enzyme that does not belong in a plant, but that makes it resistant to the Roundup herbicide. And so you can grow that corn and you can spray your field with Roundup and it kills everything but the corn. So a really efficient idea. It turns out it's dangerous, but that's a different story for a different time. But the point is, your food has DNA. So I wish that this chart also had nucleic acids, which would then, if you take off the phosphate and the base from a nucleic acid, it's sugar, right? It's deoxyribose or ribose sugars. So that would fall into our vertical category. But when you eat things that have protein, the protein is a polymer of amino acids. So if you break down the amino acid, keep in mind, or you break it down to amino acids, keep in mind what an amino acid is. It's this plus whatever various side chain it may have. Well, let's do glycine. Let's do valine. I'm feeling valine. I bet valine is purple. So if we break this down, these things look a lot like acetyl groups and pyruvates. We have a 3-carbon molecule if we break off some of those bonds, right? And then one of the things that you excrete in your urine is the ammonia from breaking down proteins. So not so different if we think of these just as carbon and oxygen and hydrogen molecules. They look a lot like pyruvates and acetyl-CoAs. And so they feed into this process as pyruvates and as acetyl-CoAs and as some of the intermediates of the citric acid cycle. They just don't start in glycolysis. If we think about what a fat is a glycerol plus the fatty acids. So the fatty acid chain just a small example would look something like this, right? So if we were to break that into smaller pieces, we would have 2 carbon molecules like acetyl-CoA. So they feed into the pathway there. If we think about the molecule to which the fatty acids are attached, which is the 3-carbon alcohol glycerol, that looks an awful lot like glyceraldehyde 3-phosphate if we put a phosphate on it. And so the point is, all of the molecules that make up the things we eat are found in this process. They just enter in different places. Now, a question that I have for you, and if you have the slide, probably you know the answer, so don't cheat. If you don't have the slide in front of you, then I want to hear your guesses. If we eat food and we store some of that food away to be used later, because we rarely eat less than we need. We usually eat much more than we need. And so we store some of that energy to help us last till the next meal. How do you think that energy is stored? After thinking about how we can break these things down, do you think, what percentage of our energy do you think is stored as glycogen? This is the sugar that's in our liver and muscle cells. It's a polymer of glucose. How much do you think? Just throw out a number. 45%. Okay, what else? Give me another guess. 10%. 18%. Says Bucky. So we got 10, 45, 18. What else? Give me one more guess. 90%. 1%. You're so close. Isn't that strange? Because glycogen is so accessible and we have so much sugar in our diet, it seems like we would be storing more of it. But glycogen is like our first reserve we call on. It's easy to convert back to blood sugar, but we just don't store a lot of long-term stuff there. We're using this constantly. So we use a high percentage of glycogen for energy, we just don't store it that way. We don't store a lot of it. What about proteins? Now that you've been burned, I hope you'll still guess. 9. 20. 25. It's like an auction. What? One more? 40. I once bought a lot of hair care products at an auction after a 5K run to raise money because it was in front of Kroger and they were celebrating the end of the race and they were auctioning off things for some charity, and I saw someone I knew. So I waved, and it turned out my wave was interpreted as a bid and I had to buy all this hairspray. What am I doing with hairspray? Anyway, that's what it felt like. 21%. I think someone was very close, right? Didn't someone say like 19 or close? Oh. What about fats? Bing. Very good. 78%. So this is just information to think about. The way that we store things. Glycogen is our first go-to thing that we can harvest energy very quickly. Fats are extremely efficient way to store energy. Those fats have a high amount of energy that can be harvested. And proteins also have a lot of energy, but we don't usually go to them, right? Usually you burn through your fat, you burn through your glycogen, and then, for example, if a person is is fasting, or if a person is in a famine-stricken region, the proteins eventually start to break down, and that's when you see really losing muscle mass and entering a dangerous state of health. Okay, I'm gonna skip that. No, I'll say something about it, but not much. This diagram is included in your book to indicate that oxygen is not always the final electron acceptor. I won't hold you responsible for this. This is more just trivia. Some organisms have evolved to not use oxygen, so they use things like nitrate as their final electron acceptor for reasons that are not clear to me. There's probably a scientific or evolutionary explanation. But the reaction still works, they just don't use oxygen. So the point of me showing you this is that not everything does it the same way, even though most things do. By far, the vast, vast majority use oxygen. Okay, so in our last minutes, let's talk about the other side of the coin, which is anaerobic respiration. This will be a nice way to end our talk today. So in anaerobic respiration, why do I feel like I'm spelling that wrong? No. Things look different when you're up at the board writing them. But in anaerobic respiration, this is respiration in the absence of oxygen, in the absence of the final electron acceptor. This can happen in our muscle cells. This can happen in organisms that are growing in the environment. So it's not just a human thing or an animal thing. And of course, it's utilized to make lots of different products. Anaerobic respiration is also not necessarily a bad thing. So, well, let's look at this reaction, then I'll finish my thought there. So in anaerobic respiration, you can't drive towards the final electron acceptor because it doesn't exist. There is no oxygen. So you never leave the process of glycolysis. And so if we think about glucose as our prime example, glucose is split, and in the process of splitting it, we result in 2 pyruvates. Remember that in glycolysis, we invest 2 ATP, we yield 4, and so our net is going to be 2. So we still get those 2 ATP, but the pyruvate doesn't go on to the next stages. Instead, the pyruvate has to be dealt with in some way. So one of the things that happens with the pyruvate is that it is converted to lactate, which is the lactic acid that builds up in your body and causes discomfort. But in the process of converting it to lactate, some of the electrons from the NADH that would be otherwise sent to oxidative phosphorylation, they don't go there, right? They can't go there. So instead, the NADH donates electrons back to pyruvate to form lactic acid. The lactic acid eventually is carried out of your cells, and you get back to your normal homeostasis. And then the NADH gets recycled and used again. So this process is not sufficient to be all the energy that you need, but it is sufficient to get you through immediate needs. And the reason that this could be beneficial is that if you pressure your body, if you push yourself aerobically or in some sort of strenuous exercises or cardio exercises, if you push yourself and your body is always running out of oxygen, it's going to adapt. And the adaptation that your body's going to make is that it will find a way to get more oxygen to your tissues. And the only way to do that is to have more vasculature, more blood vessels going into your tissues. And so you will re-engineer your vasculatory system in order to compensate for this. So that's why, and I think I probably talked about this at the beginning of this section, that's why after you train, you get better and stronger because you're changing your physiology. If you stop training, you lose that ability that you had gained and go back to whatever your normal state was. So you have to keep pushing yourself to keep improving, or at least to maintain what you have. And there are all kinds of ways that people have manipulated this or exploited this. One of the strangest ways that, I don't know if it's good or bad, I'm certainly not an exercise physiologist, but one of the things that always thought was, struck me as odd is when people restrict their oxygen with a mask. Is there a name for that? You know what I'm talking about, where it looks like people are wearing a face mask, but it is intentionally to restrict oxygen so that they put themselves under stress. It seems dangerous to me. I mean, like I said, I'm not an exercise physiologist, so maybe it's not dangerous, just seems weird. Okay. Now, of course, we've got our fitness person here, and then we've got our wine taster. Two sides of the same coin. But this is exploited in the food industry, in the beverage industry, quite a bit. I'm sure it's no surprise to you that the production of alcohol comes from fermentation. And so fermentation is anaerobic respiration that is being utilized for some purposeful end. One way that people utilize this is that some organisms produce, instead of lactic acid, ethanol. And so in the production of the small amount of ATP in anaerobic respiration, and then the pyruvate is broken down. Instead of going to lactic acid, the pyruvate yields carbon dioxide, right? So pyruvate has 3 carbons, so if 2 go into carbon dioxide, that leaves 1 left. And that is, wait, that math doesn't add up. 2 CO2s. Where's the other carbon? Where'd the other carbon come from? I may not be showing all the steps of that reaction. It doesn't add up in my head. Well, in any case, no, yes it does. Each pyruvate donates 1 carbon. That's the math. So 2 pyruvates, 2 carbons, then 2 carbons left over. Okay, sorry, had a moment there. But instead of becoming lactic acid, it becomes ethanol. And there are 2 things to notice about this. One is that carbon dioxide is still produced, and 2, ethanol is produced. So if you were to make, for example, homemade beer or homemade wine, it self-carbonates, right? If you keep that system closed and don't let the gases escape, it self-carbonates. And when it self-carbonates, eventually the yeast that is used to make wine or beer will reach a level of alcohol that causes the yeast to stop growing, or in most cases kills it. And so the yeast, in their trapped state in the bottle, produce alcohol. The alcohol poisons them, they die, they sink to the bottom. So if you make homemade beer or homemade wine, there's usually residue at the bottom, which is the dead yeast cells. Now you don't see that in production beer. Of course, most of you are not of legal drinking age, but you don't see it in production beer because in the production beer, they know no one wants to be drinking and look at the bottom of the bottle and see all this sludge. And so instead they siphon off the liquid and leave the dead yeast cells behind, and they pump CO2 back into it from just a tank. But if you were doing this on your own, that CO2 does actually occur if you don't have to pump it back in. What else do I want to say about this? Maybe that's it. Okay, that's where I want to end today. We have a few more things before it's all over, but that's a good